Swayam Prabha. Digital India. Educated India.
Then another aspect, this is very important aspect in protein structures, the solvent accessibility. The name itself tells this is divided into two, solvent and accessible. What is accessible? Reachable, like how far we can reach, right? Whether it is very close, so we can reach more, or how far we can in contact, right? So what is a solvent? With respect to a solvent, right? Okay, we have protein structures, right? If there is a solvent, right? How far the solvent can reach each atoms or each residues in a particular protein, right. So, based on that you can define the solvent accessible surface area, because the area we tell because how much area they can overlap, right. This is what we call the accessible surface area. So, you can define this as the locus of the center of the solvent molecule, and this it rolls over the van der Waals surface of the protein. So, we have the protein, different residues and they got different atoms. We can represent these atoms into overlapping spheres, right. And if we have a sphere, then we take a solvent molecule and we will roll the solvent molecule on this protein and see how far they are in contact. That contact is called the contact area, and how far the center of the solvent molecule rolls on the van der Waals surface, then that is called the accessible surface area. Usually we take a solvent, water is in which environment? Water environment, right. The protein is in water environment. So, we consider a protein's environment, then we take the solvent as water, right. So, we can take the water, right, and you can see the radius of 1.4 angstrom, right, because of the environment of any proteins. So, then what to do? So, you can see the solute molecule, what is solute molecule here? It is a protein, right. So, you can see the protein, this is a solute molecule, you can see the interlocking spheres. So, you can see this is the interlocking spheres, right, here, and we can assign the different van der Waals radii. What are the atoms present in water in protein? Carbon, nitrogen, oxygen, sulfur, right. So, based on the atoms we have the van der Waals radii. So, each atom we can have the different van der Waals radii. So, they are interlocking spheres. Now, what to do? Right, you can now roll the solvent molecule, right, on the along the envelope of this van der Waals surface, right. You can see the van der Waals surface here, right. So, this is the water molecule, right, this is the water, right, of 1.4 angstrom. So, you roll this water molecule on the van der Waals surface, and from that now you can see the radius of the surface, this is equal to r plus r the solvent molecule, and you can calculate accessible surface area of atom of radius r, right. You can see this r this is a area of the surface of a sphere of radius r, this r equal to r plus r the solvent molecule. On each point which the center of the molecule are in contact with the solute molecule, right, in contact with the solute molecule without penetrating to other atoms, right. So, if we have this water molecule which are rolling around, how far they can penetrate into each atoms, right, based on the van der Waals radius of this each atom in a protein. So, that area we call as solvent accessible surface area, right. So, we have the water molecule, now you roll on this molecule. So, this is the contact, how much you can contact, this is the contact area. Now, how much you can penetrate into the van der Waals surface, that you can get the accessible surface area, that can be that is the maximum surface area we can accessible by the solvent with the atoms. So, there are several methods, this is the principle how they estimate the solvent accessibility. There are various methods to calculate the solvent accessible surface area of each atoms to based on the different algorithms. So, I show some of the algorithms. One is access, right, this is the one first introduced in the literature in 1978, actually started in 1971, Lee and Richards, right, they proposed the concept of solvent accessibility and the program they developed in 1978. So, they used this information how the protein can be accessible by a solvent molecule. Later on they refined, right, in 1993 and they developed NXS, right, to get the accessible surface area of all atoms. Likewise, there are several other methods like ASC or DSSP or get area. Earlier we discussed the DSSP, right, for the secondary structure of proteins, right. They combine the secondary structure as well as solvent accessibility, right. At the one file you can get all the data. So, these are the some of the aspects of different methods. Why do we need different methods, right, the various aspects. So, there are several algorithms available to calculate the accessible surface area of all atoms and residues in a protein. So, the major programs are Access, which are the one which are developed early in the literature, maybe 1970s, and followed by DSSP, NXS, ASC, Criteria and so on. Currently we have several other algorithms are also available in the literature. So, we look into these different algorithms, there are some advantages and some disadvantages. For example, if you look at DSSP, so you can easily use standalone program is available, but it can give you the accessible surface area for only the residues. So, we cannot get the data for the atoms, right. So, if you take the atom wise, so it is no, but if you use the NXS, right, NXS, right, you can get the atom wise one, right, but here there is no online calculations or database, but NXS you need to get the license, right, you have to write the copy assignment and then you have to send and get the license and then you can use it. And here this will give you only the secondary structure, though information regard only the accessibility, but no secondary structure. Look at the DSSP, it will give you the ASA plus secondary structure. So, this advantage of using DSSP, you can get the secondary structure and solvent accessibility simultaneously when you run the program. So, ASC is another program, but in this case you need to edit or manipulate these files, right, depending upon your input files, right. So, you can edit, here you can say the you can change the probe radius, you can change the van der Waals radius, and source code is also available. So, this for free they will give the source code. So, in the get area, so get standalone is not available, but you can get the online. So, if you give the PDB, right, then you can get all the data, you give the polar nonpolar area, right, I can give atom wise surface area, right, but you can also use by manual editing, you can change the van der Waals radius and other parameters. So, some program you cannot change anything, right, some of case only you can get the standalone, some case only you can do the online, right, some cases you can manipulate the change your default parameters, right, and the DSSP provides both secondary structure as well as the solvent accessibility. Okay, these are the references, this is Lee and Richards. Right. So, this is the output obtained from the DSSP, right, example I show the output from the DSSP. Okay, DSSP is developed by the Caption Center, right, they published in 1983 in Biopolymers, right. So, here if you see the data, okay, here the data starts from here, right, this is the residue number, there are two numbers, one is based on the PDB file, one is actual residue number from the UniProt. So, you have the two numbers. So, here you get the amino acid sequence K, V, F, E and so on, right, and here this is the assignment of secondary structure, in this case ASCC, this is the accessible surface area. When you look into the accessible surface area, right, can you see these numbers and tell something from the accessible surface area, that which residues are buried, which residues are exposed, right. If you see the numbers, some of them are very high, for example, here this is 146 and so 114, right, these residues are exposed, right, they are at the surface. So, for example, this residue, if you see this arginine and you can see this the glutamic acid, right, these residues are at the exposed. Then some residues, for example, they are 0 and 1, right. What is the meaning of the 0 and 1? They are buried, right. So, in this case if you see this leucine alanine, so these residues are buried in the interior, fine. So, if you have any PDB code, you can download DSSP, you can standard programming is available, you can execute easily, just one line command, right, if you give the DSSP and you give the your PDB ID and if you give the output file name, you will get it, right, it is just a one line command, you can get the data for the DSSP, right. But the only disadvantage is it will give only the residue wise accessible surface area, otherwise it is very fast. This is another program, it is called the get area, here you can use the online version, but standalone is not available, but you can manipulate the probe radius and other parameters and you can get the data for all atoms, right. For example, if you want to use the PDB file, right, you can give the PDB, here I give 2 PTL dot PDB, right. So, water molecule that is 1.4. So, here if you will email address and you get the output, right, and submit for analysis, right, then you will get the data. So, if you look into this data, so each residue, this is the total surface area and they group into different atoms based on the apolar and the polar. So, amount total to 1 2, right, acceptable surface area angstrom square, apolar is about 98. Then also they classify into backbone and side chain, okay, this is the backbone and this is side chain as well as this ratio. Then based on this area they classify this is out or in, right. What is the meaning of out? This is exposed. So, values are very high, these values are very high, so they are out. So, here this is less, right, here this is less and they are in the interior of the protein. So, they can classify into either interior of the protein or at the surface, fine. So, now you can also get this area for each atoms, right, here this is for each residue. Now, here you can see even for this glutamic acid, so they have for each atoms, right, you can see the area, right, for each atom in a residue. So, this each atom type, whether this is CAB or CBB, right. So, you can see this is the backbone, right, and you can see the different atom types and you can see whether this is sulfur, right, and the aromatic, right, aliphatic. So, you can see the area for the based on the different atom types. This is overall, you can see overall polar area and the polar area, right, and this is the total area. Also there are number of atoms which are the surface, which are at the buried. So, you get all the information rugged because it will give you the data for each atom, it is easy to use. Now, the issue is most of the biologists, right, they find it difficult to interpret the data. So, in this case instead of giving numbers, if you provide a kind of figures, right, so that they can understand which residues which are at the surface and which residues are the buried, then it will be very helpful, right, to interpret the results. So, we developed the database called ASAView, right, we developed database as well as an algorithm to represent solvent acceptable surface area. It is a characteristic 2-dimensional plot, kind of spiral plot, which will give you some information, right, so that you can view the figures and then get the information, right. So, how to do that? So, here the various aspects, one is different types of atoms, different types of residues, right. So, here we considered only the residues, not different atoms, right. So, what are the different types of residues in a protein? Polar residues, non-polar residues, charged residues, right. So, we can show specifically, okay, these are charged residues or the hydrophobic residues, polar residues and so on. Then these residues, they have different surface area, some of them are less and some of them are more. You can give the circles with the different sizes, right, based on the surface area. So, we want to give the plots, right, in the form of the spirals as well as the bar chart, just give you the actual values, right, for each residue in a particular protein. You can use online plots for any PDB ID or you can upload your PDB, right, and you can get the plots. Okay, now I will show you the plot. So, here this is a plot for a particular protein. We got different colors and the different colors represent for the different types of amino acids. Likewise, the blue, right. So, you can see the blue, this is the K32, this is the positive charge residues, and the green. So, here you can see the green, this is mainly for the polar residues, right, based on the notations, and you can see the gray is mainly for the hydrophobic residues, valine or alanine, and the yellow is mainly for these cysteine residues. I do not find any cysteine here. And you can see the size, some of them are very small and some of them are very big, that represent the relative acceptable surface area of each residues. From this one, can we guess which residues prefer to be the interior, which residues are in the surface? Mainly we see the non-polar residues, okay, see the stretch of these grey colors inside the core. So, most of the hydrophobic residues, right, these are the hydrophobic residues, right, which are mainly at the interior of the core. In the surface, if you see mainly the polar residues, and you can see the charge residues, positive charge and the negative charge residues, right. You can see the distribution of these residues, right, at the interior of the protein or at the surface. This is another plot, this is a bar plot which give you the value for each residue. For example, here this starting from the 1 to n, that n terminal to the C terminal will give the data. If you look into this plot, here also you can see they continuously not with the high ASA or low ASA. Right. For example, if you see this residue and here you can see they are distributed differently in the sequence, right, here these residues, but when you look into this number they are buried in the interior. So, comparing this plot and this plot, right, some residues which are far away in the sequence they are come close to each other, right, and they are placed at the interior of the protein. This also supports this hydrophobic model, right. So, that they collapse and then they form the core, the hydrophobic residues, and the other polar residues which are at the surrounded by these hydrophobic residues to make the interactions, right, with the water as well as with the other residues. So, here and there if you see these hydrophobic residues, but they are very less ASA, but they are mainly in the interior of the protein. So, we have the values, we can plot, either you can make spiral plot or you can make the bar chart to see how these residues are distributed, right, based on accessible surface area. So, we have the web server. So, you can it can take any PDB ID, right, so you can get the PDB ID. So, if you can get plot, right, then you can see that what are the types of plots you require. So, depending upon which one you want, the PDF version or the any relative values, whatever you need. So, right, you can see the graph, this spiral plot, and you can see the bar chart. In addition, we use the actual values. Okay, if you see this one, this is actual surface area. This is output from the DSSP. We give the link so that they can the users can use and they can see the exact values for each residues in their particular protein of interest, right. Fine. Here this is the value for each residues, right. This is only the ASA values. So, now if you look into these different residues, for example, if you compare glycine and the lysine, how many atoms in glycine? 4. So, only where the heavy atoms, only 4, only the main chain atoms. If you take alanine, it is 5. If you take arginine or tryptophan, right, you have more number of atoms. So, when you do the residue wise accessible surface area, they add up, they do it for all the atoms and finally add up, then they give for the residue. In this case, there is a bias of these residues, some of them are small, some of them are big, right. In this case, we need to normalize how far the residue is accessible, right. In this case, you can calculate the percentage accessibility, that is the ratio between the accessible surface area, right, which are compared to 3D structures as well as the extended state. What is the meaning of extended state? The maximum it can be accessible, right. In this case, there are two different ways. One is you can take this tripeptide or trinucleotide, right, and you can use this concept to get the accessible surface area. And the most easiest one is they take the conformation glycine x glycine or alanine x alanine. Why they use glycine x glycine?
It does not have side chain.
Yeah, glycine has no side chain. So, in this case, if it is central residue is any residue, this is highly accessible because neighboring two residues they do not have any side chain. So, look at the glycine x glycine in all the proteins and see the conformation, and from that they can take either the highest value or they can take the maximum the average value, right. From that they will see this will be the probable accessible surface area for any residue when they are extended state, right. These are the values for the 20 residues, and as you see glycine has the lowest one, and you can see the tryptophan or the arginine, right, you can see the highest values. Now you can get the ratio, one from the 3D structures, you run the program and any of the programs. What are the programs we discussed? NXS, Access, AAC, Get Area, right, or DSSP. You get it for the folded state and extended state values we know, then you divide that, right, then you will get the percentage accessibility, right, the percentage ASA, right. So, that is ASA in the 3D structure, right, and the ASA in the extended state. From this you can calculate the percentage, right, multiply 100. So, then the value is less than 5%, then we call these residues are buried. What is the meaning of buried? They are in the interior core, right. For example, if you see this figure, okay, this is the buried, and you can put the value of 5 to 20%, right, this is partially buried, and 20 to 50 as partially exposed and more than 50 as exposed, This is the general values we use in the literature, but this is not very strict. Here also you can change your cutoff based on the type of applications you use. Generally, if it is less than 5%, we use buried. Some cases they use 2%, right, or that is changeable, right. So, 5 to 20 is partially buried and 20 to 50 is partially exposed and 50% is exposed. So, this is how we can get the acceptable surface area, convert it into percentage, and we can do it for the analysis depending upon the location of each residue. So, we summarize what are the various aspects we discussed today.
Structure classes.
Yeah, different structure classes. What are different structure classes?
Scope and
Right, all alpha, all beta, alpha plus beta, alpha by beta. Right, what are different databases which can give this information? Cath and scope. Right, then we consider contact maps. What is the contact map? This is a plot connecting the contact between two residues in protein structures. Right, in construct any space, we can define a distance and different atoms, and if they are in contact, we put a dot. So, this will give you the 2D representation of the contacts between residues in protein structures, right. Right. So, from that we can define long range contact or short range contact or the medium range contacts depending upon the residues which are close in space, but how far they are distant in the sequence. So, then we discussed about solvent accessibility, right, how far each residue is accessible to a solvent, right. There are different programs to get the solvent accessibility, and we can represent in a pictorial view. So, for example, which is the program to get the pictorial view? ASA view, right, and to get the percentage accessibility. So, get the surface area at the 3D structures, and you can do it for the extended state and get the ratio, right, then you will get the percentage ASA. Based on that, we can classify into buried or partially exposed or exposed, right. So, you can also derive various other parameters, say contact order or long range order, and the buriedness, preference of residues to be the interface, and the free energy and different types of interactions, right, and how about the electrostatic or the residues which can form disulfide bridges, right. So, there are various features, right, the properties you can derive from the protein 3D structures, right, and we will look into details, right, in the following classes. Thank you for your kind attention.